There are many similarities in fungicide resistance, insecticide resistance, and herbicide resistance. The terminology is a little bit different among plant pathology, entomology, and weed science, but the concepts are very similar. In this presentation, we use the terms insecticide resistance and pesticide resistance interchangeably. In entomology, we deal with insecticides and acaricides or miticides, but sometimes it's easier just to say pesticide resistance when referring to these, even though pesticides refer to a broader range of materials. What is pesticide resistance? It is a decreased susceptibility of a pest population to a pesticide that was previously effective at controlling the pest. The pesticide is no longer working as well. A more technical definition of insecticide resistance is a heritable change in the sensitivity of a pest population that is reflected in the repeated failure of a product to achieve the expected level of control when used according to the label recommendation for that pest species. What is a heritable change? It is a change in the genes of individuals in the present generation that is passed on to the next generation. Resistance develops in individuals of a population. A population is a group of individuals of the same species living at a given time and at a given place. For example, 2 spotted spider mites on corn leaves in one part of Tulare County is a pest population. That population may be different from 2 spotted spider mites on corn leaves living in another part of Tulare County. Different populations can have different traits and characteristics, such as different resistance levels to various toxicants. Resistance happens at the population level. Insecticide resistance is not a new occurrence. DDT started the era of synthetic insecticides when it was discovered in 1939. Paul Mueller discovered the properties of DDT and won the Nobel Prize in Physiology or Medicine 1948. In 1947, houseflies developed resistance to DDT, and later many other insects and mites became resistant to this pesticide. So resistance in insects is certainly not anything new, and furthermore, it is not something that only happens to synthetic compounds. In 1914, scale insects developed resistance to lime sulfur, an inorganic compound. How does resistance develop? Let's say you have a population of spotted-winged Drosophila. They all look the same out in the field, but the boxes around the 3 individuals indicate that there is something unique about them. You can't see it, but they may differ metabolically, behaviorally, or structurally. They are all the same species, but these 3 differ in some way. You spray these spotted-winged Drosophila, and what happens? You've killed most of the normal ones, but it didn't touch the 3 that were different in some way. So what is going to happen to the next generation? Those 3 different individuals reproduce, and you see that you have 11 of them now. You also had 1 normal one, and that one produces another normal one. So your population in this example has shifted primarily from the normal individuals to these different sorts of individuals. What happens if you spray again? You were left with the unique ones. It killed some of the unique ones, but you don't have any of the normal ones left. If you go through another generation and spray again, nothing happens. The population has switched from primarily normal individuals to 100% of the ones with some unique characteristic, and that unique characteristic is resistance. The insecticide was the selection pressure, and it selected for these different individuals, but the bottom line is the population you started out with had genetic flexibility that created different individuals. You didn't create the genes, they were already there, but the insecticide selected for the genes in these different individuals that gave them resistance, and now your population consists of 100% of the unique ones. Genetics and selection pressure are the 2 most important factors that influence insecticide resistance. There is genetic variability in the individuals of the insect population. Then selection pressure, intensive application of insecticides, brings those genetic differences to the forefront by eliminating the susceptible individuals and causes the unique individuals to become the majority of the population. What affects how fast insecticide resistance develops in insects? There are a number of different factors. The types of resistance genes, how many are involved, and are those alleles dominant or recessive? If they are recessive and they mate with susceptible individuals, then resistance doesn't manifest itself in the next generation. If the genes are dominant, then even when the resistant individuals mate with susceptible ones, their children can resist the insecticide. How many generations does a pest have per year? Pests that have many generations tend to develop resistance faster than those with a single generation per year. Citrus thrips with 8 generations per year developed resistance to OPs in the 1980s. California red scale with 4 generations per year developed resistance in the 1990s, and Citricola scale with 1 generation per year developed resistance in the 2000s. How mobile is the pest? Mobile pests that are highly motile and mate with susceptible individuals are less likely to develop resistance than pests that are immobile. Once a population of California red scale developed resistance to OPs, it tended not to lose it because there is very little influx of susceptible individuals into an orchard. The males don't fly very far. The worst-case scenario is many greenhouse insects and mites. There are few susceptible individuals coming into the house to breed with. They have rapid life cycles, often dominant resistance. Pesticide applications are applied frequently in greenhouses because there is a low tolerance for damage. Resistance can develop very fast in greenhouses. The persistence of the pesticide residues and the overall selection pressure imposed is another important factor. The more you spray and the more persistent the pesticide, the quicker the selection of resistance. Insecticide resistance is different from insecticide tolerance. Tolerance is the natural ability of a population to withstand the toxic effect of an insecticide. Not all insecticides kill all insects from all species. For example, if you use imidacloprid and try to kill beet armyworm, it isn't going to kill these larvae very well. Never did, never has, never will. Beet armyworm as a species is naturally insensitive or tolerant to it. The biocontrol agent green lacewing can walk through pyrethroids and is unaffected because it naturally has very high esterase enzymes. How common is resistance worldwide? There are about 600 species of insects and mites that are known to have developed some level of pesticide resistance. There are literally dozens of insecticides and miticides, to which 2 spotted spider mites, diamondback moth, Colorado potato beetle, and sweet potato whitefly have developed resistance worldwide. Colorado potato beetle is found in most other states except California, and this graph shows the number of different active ingredients that Colorado potato beetle has developed resistance to. You can see the steady progression over the years. There is no stable insecticide management for this insect. If you have an insecticide that works for 1 year, chances are that it won't work the next year or at least the year after that. This insect just has the perfect set of characteristics that allows the insecticide to go right through. Insecticide resistance develops in agricultural pests, medical and veterinary pests, and also in natural enemies. We don't mind when natural enemies such as predators and parasites develop resistance because it allows them to survive the pesticide and help control the pests. But resistance in natural enemies doesn't happen as often as the pests. What taxa are involved in resistance development? Many species of Diptera, flies and mosquitoes, Lepidoptera, caterpillars, Acari, mites, Coleoptera, beetles, and Hemiptera, sucking insects, have developed resistance to pesticides. Why is this so common in insects? Insects have been and still are extremely adaptable organisms. There are about 900,000 different species of insects in the world, representing about 80% of the world's species. They are survivors, they're adaptable, and they evolve. Insecticides are organized into classes, such as organophosphates, carbamates, pyrethroids, neonicotinoids, and others. Within each of these classes, they share a similar chemical structure and a common mode of action. That is, all the insecticides within a group have a similar way of killing the insect. There are currently 28 different groupings of insecticides or miticides. Most of them attack the nerves and/or muscle system in the insect. That has been one of the most fruitful ways to kill insects. Design insecticides to attack the nervous system. But insecticides have also been designed to attack their molting process, respiration, and midgut. And with some insecticides, we don't know how they work. The Insecticide Resistance Action Committee, IRAC, categorizes pesticides into these classes. Chemical companies are always trying to develop insecticides that fall into new groups and work by a different mode of action to get around some of the resistance issues. Group 3 insecticides are the pyrethroids. All of these have the same mode of action. That is, they attack the sodium channel in the insect nervous system. Because they attack the same part of the insect, if an insect population develops resistance to one of the materials in the group, it will be resistant to all of the pyrethroids. The mode of action is the process by which an insecticide kills an insect or inhibits its growth. The target site is the exact location where the insecticide works. If the mode of action affects the nervous system, the target site might interfere with one certain enzyme within the nervous system. The mode of action and target site are often used interchangeably, but they do differ a bit in what they mean. As said previously, there are 28 different modes of action. 11 of them work at the nervous system or muscle system. 7 of them affect growth and development of the insect. These are the insect growth regulators that prevent egg hatch or molting by larvae and pupae, or prevent the development of the chitin exoskeleton. The muscle system mode of action is the latest so-called hot area of development. Ryanodine receptor modulators is a fairly new mode of action on the market for the last several years. Coragen and Verimark are 2 examples. They are called diamides. There are 6 different classes of insecticides that work at the insect respiration or energy systems. Then there are some that work in the midgut of the insect. Those are primarily the bacillus or Bt products. There are several ways that insects and mites develop resistance to insecticides. One is called metabolic resistance. Resistant insects can detoxify or break down the toxin by increasing the number or type of enzymes they have in their bodies. These enzymes then bind to the toxin and prevent it from poisoning the insect. This is the most common type of resistance in insects, and it presents the greatest challenge for resistance management. Another type of resistance is behavioral resistance. One portion of the population behaves differently than another portion and is selected out by the pesticide. Mosquitoes are adept at this. Some populations have developed a preference to rest outdoors because the portion that naturally rested indoors was eliminated. Houseflies can be selected to do the very same thing. Altered target site resistance is another general way that insects can develop resistance. The site where the toxin usually binds in the insect becomes modified to reduce the insecticide's effect. Finally, penetration resistance is another one. Insects with a thicker cuticle survive because it slows the insecticide from penetrating the body of that insect. Now let's go through a couple of different modes of action to show how resistance has developed. Organophosphates and carbamates affect the insect nervous system. The diagram shows the insect nervous system. You can see the brain. They have a small brain, central nerve cord, and small axons coming off to regulate the movement of the wings and legs. The one difference between the insect nervous system and the human nervous system is that the insect one exists in several units. The gap between nerve axons is called a synapse. In the insect nervous system, a signal comes down the axon, then it has to jump the synapse, and the signal goes to the next axon, and so on. So the insect nerve is not just one cord or complete line. It's a series of subunits. When a signal reaches the synapse, the compound acetylcholine is released. This is a neurotransmitter, a messenger, or signal. That neurotransmitter jumps the synapse and plugs into the receptors on the next side, and the signal goes on. The signal may be to move the leg or wing. After the signal has been transmitted to the other side, the enzyme called acetylcholinesterase resets the synapse and turns the signal off. If an OP or carbamate insecticide is in the insect system, it binds with the acetylcholinesterase so the synapse can't reset and the signal keeps on transmitting across the synapse. The nerve stays in the excited transmitted state and the insect can't function normally. When the insect is exposed to the insecticide, the nerves continue to fire and the insect has tremors, convulsions, and paralysis. In resistant individuals, they have increased levels of esterases, some for the pesticide and some for normal function of the synapse, so that it stops firing and the insect returns to a normal state. Another group of insecticides is the neonicotinoids. These also affect the nervous system, but in a different way. Acetylcholine is the chemical that crosses the synapse and causes the signal to be transmitted. Neonicotinoids plug into these receptor sites and block the acetylcholine. The acetylcholinesterase cannot reset the synapse because it can't break the bond between the neonicotinoid and the receptor, and the signal continues to be transmitted, i.e., the insect continues to move its legs, for instance. Resistance in neonicotinoids has been very slow to develop. Scientists have found an enzyme detoxification mechanism and some target site protein changes in insects that are known to be resistant to the neonicotinoids. This suggests that multiple mechanisms and multiple genes are involved, but resistance has been slow to occur, suggesting that genetics are not dominant. Insect growth regulators are compounds that occur naturally in insects and not humans. Scientists were able to mimic those, reproduce synthetic ones in the laboratory, and take advantage of weak points in the developmental process of the insect. There are 3 or 4 classes of IGRs. There are chitin synthesis inhibitors that disrupt the skeleton production of the insect, and there are hormonal IGRs such as juvenile hormone mimics that keep the insect juvenile, never reaching adulthood, and molting hormone agonists that prevent the insect from growing through molting. The insect body contains a substance called chitin. We have our skeleton inside our body, but insects have their skeleton on the outside of their body, basically a suit of armor. That skeleton in the insect is composed largely of chitin, which is a very tough, durable type of product. The only way that an insect larva or nymph can get larger is to shed that skeleton. When it does that, we call it molting. Each time an insect molts, the exoskeleton gets larger, and the new exoskeleton hardens up and contains chitin. So if the insect has been exposed to a chitin synthesis inhibitor, it molts, but when it tries to form its new exoskeleton, it can't, and the insect contents ooze out. There is also chitin in insect eggs, so there is some activity killing insect eggs as well. Juvenile hormone mimic is one of the hormonal types of IGRs. Juvenile hormone is a compound that when it is at high levels in an insect larva and the larva molts, it progresses to another larval stage. When it wants to molt from a larva to a pupa, JH must be at a low level in the insect. This is how the insect is designed. If you've exposed the insect to artificial juvenile hormone when everything else in the insect is telling it to go to the pupal stage, the insect gets cross signals and it could stay a larva or only partially develop into a pupa and die. Resistance to insect growth regulators has been fairly slow to develop since these are compounds that naturally occur in these insects. California red scale has been treated with the insect growth regulator pyriproxyfen on a yearly basis since 1998 in many orchards, but resistance has been difficult to document in the laboratory. The newest group of insecticides to come on the market, group 28, are the diamides. They bind to ryanodine receptors in the muscles, activating them to release stored calcium. The calcium is needed for muscle contraction, and without it, the insect becomes paralyzed. There has been resistance to ryanodine insecticides found in diamondback moth in several Asian countries. The resistance appears to be due to a change in the target site. So how do you know if the insects have resistance or not? In the past, you would have used a handheld microapplicator, filled it with insecticide, and tested laboratory-reared insects with different doses. This was very laborious. You needed a large number of insects. Another way it is commonly done is looking at field trial efficacy. If you look at these data on blue alfalfa aphids from the Imperial Valley, you see the number of aphids in the check plots versus 5 commonly used insecticides. The insecticides are not killing all the insects, but it is hard to say if it is due to resistance. Another example with blue alfalfa aphid. Looking at the field data, you might say there is some resistance with Lorsban on the March 28 date, but with all other factors involved, it is hard to interpret field data. The third way to determine if resistance is occurring is with laboratory bioassays. There are currently hundreds of bioassay methods. It depends on insect species or what system you're working in. It involves dipping leaves, dipping fruit, or coating inert substrates such as petri dishes with different doses of insecticides. Other examples are coating bottles or plastic bags. Diet incorporation is another type of bioassay. In these bioassays, you place the insects on the pesticide-treated surface and determine if they live or die after an interval of time such as hours, days, or weeks. These are relatively inexpensive. The results depend on the method used, and you need a source of insects. Biochemical techniques can be used if the resistance has been documented to be due to an increase or change in the enzyme. Molecular or DNA assays, or ELISA, can measure an enzyme or other compound in the insect to determine if it is resistant. California red scales that are resistant to OPs have higher esterase enzymes. The level of esterase enzymes can be measured by grinding up individual scales and mixing them with chemicals that turn dark in the presence of these enzymes, and the proportion of resistant individuals in a population determined. What do you do when you collect these data? Scientists measure the resistance in a population. They look at the response of the insect to a series of concentrations of pesticides and calculate the LD50, which is the concentration of pesticide that kills 50% of the population. The resistance ratio is found by dividing the LD50 of the resistant population by the LD50 of the susceptible population. In this example, it takes about 2 ppm chlorpyrifos to kill a susceptible California red scale, but it takes about 70 times that amount to kill a resistant population of scales, so the resistance ratio is 70x. Here's an example with the rice water weevil. There have been reports by PCAs that the compound lambda-cyhalothrin was no longer working. Weevils tested from 2 different sites had LD50 values of 0.096 and 0.32 ppm. This suggested that there was a 3 to 4-fold resistance between the 2 sites. But you can't tell if there is resistance occurring unless you have something to compare it with before the chemical was used heavily. Fortunately, there was some data from 1999 when this product was first put on the market that it suggested that it took 1.5 ppm to kill this insect. Insects were more susceptible to the compound than back in 1999. This shows that when these compounds first come on the market, you really need to develop baseline data. A discriminating dose or concentration is another way you can develop resistance data. You test a series of different doses and develop a line that shows when you kill more than 99% of individuals and use that dosage to discriminate between susceptible and resistant individuals. If they survive it, then they must be resistant. So how do you manage resistance for insecticides? The goal is to reduce selection pressure. How do you do that? Avoid applying insecticides is of course the best way, but in most cases is not practical. Use non-chemical management tactics as much as possible, such as preserving or augmenting beneficial organisms. Minimizing the number of applications is another way. Rotate insecticide modes of action. Change it up on the insect. So if you've used a Group 18 product, next time use Group 20. Unfortunately, the number of products and the number of modes of action are limited for most pests. Using a high label rate tends to select resistance slower than a low label rate because it does not allow many of the insects to survive. If you look at insecticide labels, they tell you what group the insecticide falls into, so you know right away what mode of action the group falls in. Use good IPM practices. Monitor pests. Use research-based sampling procedures to determine if pesticides are necessary, based on action or economic thresholds. Use the best application timing, when pests are the most susceptible. Employ appropriate control measures. Effective IPM-based programs will include insecticides, cultural practices, biological control, predators and parasites, mechanical control, and sanitation. Select and use insecticides wisely. If repeated applications of pesticides are necessary, alternate insecticides with different modes of action against the pest so that no more than 2 consecutive applications are made with the same mode of action. The insecticides used in a rotation or tank mix must be active against the target pest. Follow label directions for the proper application method and rate. There are trade-offs in using low rates to preserve natural enemies and increasing the selection pressure for resistance. Minimize the use of long residual insecticides because that extends the period of selection pressure. When persistent pesticides must be used, consider where they can be used in a rotation scheme to provide the control needed and with a minimum length of exposure. Select insecticides that are least damaging to populations of natural enemies. When feasible, spot treat or leave unsprayed areas within treated fields or adjacent refuge fields. The pesticide-susceptible individuals in the untreated areas will interbreed with resistant ones and dilute the resistant genes in the population. Keep good records of insecticide use to aid in planning for future years. Tank mixes or premixes are becoming more and more common in entomology and other disciplines. From the entomology standpoint, premixes aren't a good thing. A PCA or grower may take insecticide A from group 3 and insecticide B from group 10, and using the appropriate rates, do a mix. But if you are buying a premix in a jug, many times the rates are not optimal for both products, and one of the pesticides in the mix may not be targeted for the pest you intend to control. It is better to purchase the insecticides as individuals and mix them for the target pest you are interested in controlling. In addition, it is better to apply 2 pesticides days or weeks apart in a rotation. With 2 separate applications, you cover more of the host plant and extend the effect of the insecticide compared to when you apply the 2 chemicals mixed together in one application. In weed science, everything is tank mixes. You control a broad spectrum of weeds. You mix herbicides that have a different mode of action but similar activity on the same species. You eliminate the possibility to develop resistance on each of those, but can eventually have the possibility of multiple resistance. There are 4 scenarios of rotation of pesticides seen in this slide. Scenario 1. If there is no rotation of insecticides, selection pressure will be very fast. Scenario 2. If there is rotation within generations, the insect is being stressed by 2 different chemicals in the same generation and resistance is slowed. Better yet, Scenario 3 is rotating between generations to allow recovery of susceptibility over an entire generation and should slow resistance even more than Scenario 2. Scenario 4 is rotating both within and between generations. This is the ideal. Often there is a cost to the insect to develop resistance. If they have to make more enzymes or different enzymes to withstand the pesticide, they may produce fewer or smaller progeny or live a shorter time. This is important because when the sprays or selection pressure stops, often the susceptible individuals are more fit and the population can gradually shift back to less resistant. Let's look at some success stories. Spider mites and abamectin in cotton. Abamectin has been used for 20+ years in California. 2 spotted spider mites have developed resistance very rapidly to many materials in many areas and crops. Abamectin still works to control mites and is still a top used product in California cotton. There was a blip of resistance seen in the 1990s in a bioassay, but it wasn't seen again. Why is this so successful? Abamectin used to be a very expensive product, so that limited use to some extent. The treatments in the California cotton system were used on a timing manner. They were applied in mid-June, not waiting until you have a large population of mites or when the cotton is huge to inhibit good coverage. A lot of cultural controls are also used in cotton to control spider mites, so that has kept resistance down. Contrasting this with the situation in southern cotton in Louisiana and Mississippi. They've had spider mites for 5 years now. They started using abamectin and other products, but now have resistance to abamectin. They didn't use full rates and only did cheaper treatments. They didn't monitor their fields, so only applied the product when they had a full-blown problem. Their lack of management of the insecticide has cost them. A very similar success story is with the neonicotinoids for melons and vegetable crops for whiteflies in the Southern California desert areas. Whiteflies tend to develop resistance very fast. These are soil-applied long-lived treatments, so it can favor resistance to develop very fast. After using these products for 20 years, they are still effective at keeping whiteflies under control in this area. BT cotton, or genetically modified cotton for Lepidoptera, is another success story. This is not in California, but in the rest of the U.S. It is highly regulated, highly researched. They have regulations in place to produce susceptible individuals. There are other technologies in the system. There has been some slight slippage in susceptibility, but still highly effective with no real resistance seen. Contrasting this with a similar situation in India. They have the same products, but after about 5 or 6 years, they've got full-blown resistance in their Lepidoptera insects. They had the same rules as in the U.S., but the rules were not enforced. Whitefly management in Arizona cotton, melons, and vegetable crops. This is a highly researched area. They do annual monitoring of resistance levels. If they saw resistance starting to develop, they would know it. There is cooperation among the different industries and different commodities, public and private. There is a limited number of effective insecticides and they are managed very well. After 20 years, the system is still in place and doing very well. So research, cooperation, and staying on top of the situation has helped to keep resistance down. So what does it take to maintain susceptibility in some of these problematic situations? Good science, good cooperation, and communication within the industries and among the PCAs. Well-trained people in the field and good observational skills are key. Many times, the growers and the PCAs are the first people to see it and communicate back to the researchers. Continuing to develop new management technologies and educating the general public is important to allow these things to be used. This concludes the presentation on insecticide resistance.